Hi, I'm Hannah, and in this video we're going to talk about another process involved in aging, cellular senescence. In my previous video, I introduced a type of protein called an apoptosome. These are produced in response to cell death stimuli as part of a cell's self-destruct mechanism. And it is important that our cells can self-regulate because they are exposed to a lot of stresses throughout our lives, making it inevitable that some cells lose their proper function. But what happens when a cell no longer responds to these signals? Senescent cells are cells that no longer have the ability to divide, and unfortunately, they also are often immune to apoptosis. This already sounds kind of bad for us, doesn't it? But before we explore how this impacts aging, let's define senescence a little better. A senescent cell is defined as one that can no longer undergo mitosis, meaning that it can no longer divide. And there's strong evidence that this was evolutionarily beneficial, like apoptosis, in preventing precancerous cells from forming. However, a well-functioning cell would ideally always be able to divide because we need new cells to repair tissue damage. But unfortunately, normal human cells will in nature always reach a senescent point. This phenomena, that normal human cells can only divide a limited number of times, was discovered in the 1960s. The experimental design is pretty simple, actually. Dr. Leonard Hayflick, an American anatomist, put human fibroblasts, essentially skin cells, in a petri dish and observed their growth. It was found that, despite the unlimited number of nutrients in their environment, the cells could only divide a maximum of 40 to 60 times. This is coined the Hayflick limit. Causes of senescence are numerous, but in the case of the Hayflick test cells that have no external stressors, it is clear that some of this must be internal. So, a cell's programmed obsolescence is somewhat explained by the existence of, yes, you guessed it, telomeres. Most of us are familiar with the basic structure of DNA, the double helix. In multicellular organisms, DNA is linear, opposed to most bacteria, which have circular DNA. Now, I find the term linear to be a bit misleading of a description because for most of a cell's life, DNA is kept in a form called chromatin. But what is meant by linear is that there are two definite ends. At the end of each line of DNA is a repetitive sequence of DNA that is the telomere. Because the telomere is only single-stranded, it curls at the end like this. The length of this curly guy plays a large role in determining a cell's Hayflick limit. So far, the most important thing to know about telomeres and aging is that they're at the ends of our strands of DNA and that over repetitive replication, they shorten. And when the telomeres get too short for further DNA replication, senescence is signaled and the cell can no longer divide. After the discovery of telomeres, it was clear that further insight into why they shorten during DNA replication would clue how to prevent or maybe reverse this process. The process of telomere shortening can be described by the following reasoning. When our linear DNA undergoes replication, the double strand is broken at a point called a replication fork. Our DNA is so long that, actually, this needs to be broken at many points, which are all worked on simultaneously, so our cells can divide at a reasonable pace. There are many molecules involved in this process, proteins that open DNA, proteins that keep it open, and so on, but our key player is DNA polymerase. DNA polymerase can only add nucleotides, the building blocks of DNA, in one direction. The actual replication by DNA polymerase is initiated at a point called the origin of replication. Then, with some help from other proteins, it moves along the leading strand, continuously adding nucleotides. But when it gets to the break of the gene, where the other half is, we realize it can no longer keep adding continuously. Instead, with the help of some little RNA pieces called primers, the polymerase adds the nucleotides in chunks. RNA from these primers are converted to DNA by DNA polymerase delta. This process works well for replicating DNA in the middle of the strand. The trouble is that the last primer place marks the end of what will be replicated. DNA polymerase delta is finicky. It won't convert just any RNA sequence to DNA. It will only convert it if there is DNA in front of it. Thus, the last primer will not be converted to DNA, and the sequence there is lost. This means that replicated DNA has shorter telomeres than the original. Let's now take a step back. This whole process seems really complicated. And do we really need to care about the loss of telomeres? So what if my cells are senescent and can't divide anymore? What harm does that really do? Well, senescent cells have many phenotypical markers, but the most important in regards to aging is that because their gene expression is damaged, they secrete toxic concentrations of what were previously well-regulated molecules. These secretions result in chronic inflammation that cause pain and tissue degradation. For example, inflammation at the joints cause arthritis, and inflammation in the heart and lungs causes shortness of breath. And since many senescent cells are immune to apoptotic signals, we can accumulate inflammation-inducing cells as we age. At first, we might find this information disheartening, but we should not lose our determination. We must remind ourselves that our body is literally a giant reaction chamber, and that the determined chemist with the right tools can control what happens on the inside. It is thus only right to conclude this video by presenting some of the strategies that biochemists are currently using to prevent and treat cell senescence. One option that falls out of our analysis of DNA replication is to lengthen telomeres themselves. In fact, our hard work investigating this system has given us a lead. Telomerase is naturally found in humans and other organisms and is capable of extending telomere length. However, it is not active in most human cells. You have probably heard that some animals are expected to live almost indefinitely in the absence of predators. Thus, the magical longevity of the lobster can be attributed to telomerase. The big question then is if telomerase can be effectively integrated into human cell systems. Telomerase gene therapy has been shown to expand lifespan and reduce cancer in mice, and one company, Libella Gene Therapeutics, is currently recruiting for human clinical trials. Should human trials prove successful, this would be a monumental step in preventing aging. An alternative strategy is to target and destroy senescent cells themselves. This requires the pharmaceutical to both differentiate senescent cells from functioning cells and efficiently deliver the therapeutic agent. Nanoparticle delivery systems have shown great potential as they can be coded to bind favorably to overexpressed compounds in the senescent cells, and they are loaded with therapeutic agent. Some products targeting and killing senescent cells are approaching the clinical trial stage. The Division on Senescence and Inflammation at Buck Institute created Unity Biotechnology just for this. Their company is very promising and currently backed by Jeff Bezos. Another company, Oisen Biotechnologies, are working towards a delivery system for their therapeutics, which have shown positive test results on laboratory mice cells. If you watch this whole video, we saw that shortening of telomeres through repeated cell division eventually leads to senescence. While senescent cells currently accumulate as we age, causing inflammation, scientists are working on solutions right now to restore telomeres and remove senescent cells from our bodies. Thanks for watching.